Hello, AP Biology students. We're going to go over the 12th section in Unit 4. And in this section, we're going to talk about something called changes in chromosome number. The image that you're looking at here is called a karyotype, and this is a visual display of chromosomes arranged by shape, size, and banding pattern. Basically, what scientists can do is they can take chromosomes out of a cell at a certain point throughout cell division and stain them with a certain dye. This dye will stain the chromosomes in different colors, usually dark or lighter bands. Now, it's really important to remember that chromosomes don't actually look like this organized in your cells. It's also important to remember that the chromosomes aren't usually going to be condensed like you see here. They're going to be unraveled or unwound so the cell can read the DNA. What we're able to use these karyotypes for is to see if there is a difference in the number of chromosomes than the average cell would have. For humans, remember, we have a diploid number of 46. There are 46 chromosomes because there are 23 pairs of chromosomes. But sometimes some individuals will inherit more or less of these chromosomes. So these individuals will have more than 46 or less than 46. This is called non-disjunction, or the failure of chromosomes to separate properly during meiosis. When this happens, you're going to get sex cells with a different number of chromosomes. As you can see, normal division is going to take those two chromosomes and divide them where each resulting cell only has one of them. During non-disjunction, either during meiosis 1 or meiosis 2, you're going to get cells with an abnormal number of chromosomes. You can see this one has one more, this one has one less, and you can see again that happens on that side as well. Non-disjunction occurs either when the body is making sperm or egg cells and creates a sex cell with more or less chromosomes than 23 for humans at least. Now, what we call the average number of chromosomes in a species is called euploidy. Aneuploidy is the change in that number of chromosomes, either more or less. So for humans, it could be like 47, 45 for aneuploidy, but our euploidy number is 46. When a zygote or when a person receives one less chromosome than what is expected, that euploidy number, we call that monosomy. This is when there is a homologous pair of chromosomes that's missing one of the pairs. When there is an extra chromosome, we call it trisomy. This is again when there should be an homologous pair of chromosomes, but there is an extra in that pairing. Now, you might think in either case, well, it's really not that bad because with monosomy, we still at least have one of those chromosomes, so at least we have one set of directions. Or within trisomy, we just have an extra set of directions on how to make things. However, in cells and just in life in general, it's important to have the euploidy number of chromosomes because it's important to have the just right amount of genetic information. I'm going to give a little bit of a warning here. I like to just kind of pause and remind my students when I go over this content that we are going to be talking about examples of individuals that have certain conditions or syndromes. I like to joke around with my students. I like to put up memes. I like just to have fun in class, but this is a time where I have to remind the students where this is a serious topic that we need to talk about and show respect to those individuals. I think that this class really shows that people inherit these conditions or certain circumstances in life that they really didn't ask for, but they are just living that life. So it's important for us to remember that we are focused on learning about these conditions so we can have a better just outlook on life and understand where somebody is coming from. From this point on, I'm going to show you 3 examples of the more frequent trisomies that we see in humans. Typically, all of the other trisomies are not even going to be born because there are such problems when some zygote is going to inherit an extra chromosome. They're usually miscarried early on. Every case, though, is different. There's just, a lot of just variation out there within even what I'm talking about. So just understand there is more to this topic than what I go over. Our first example of a trisomy is trisomy 13. You can see here is our karyotype, and there are 3 chromosomes when there should be 2. This is also known as Pateau syndrome, and there are major circulatory, eye, and brain defects in the individuals that are born with trisomy 13. Depending on the set of data you look at, it's about 1 in 5,000 live births, and in these cases, children rarely live more than a few months or years. Typically, individuals who have inherited trisomy 13 won't live to adulthood. Our next example of a trisomy that can occur in an individual is trisomy 18. You can see the 3 chromosomes in the 18th pairing of chromosomes in this karyotype. This syndrome is also known as Edwards syndrome, and it's a little bit more rare. It's about 1 in 10,000 births. It affects almost all of the organs, especially the heart. And again, children rarely live more than a few months or years. Our last example is the only example that we typically see where the individual live to adulthood, and that is trisomy 21. Individuals that have trisomy 21, this is also known as Down syndrome. This is the most common type of trisomy. It's about 1 in 700 live births, and they can also have physical and mental abnormalities. Again, there is a variation within the severity of each of these syndromes, and this is just to better understand the inheritance patterns that lead to individuals that may have these syndromes and what causes and effects they may see.